Okay, and our next speaker is Wan Sin Heng, who is here from the University of Tasmania. Thank you.
So I just click next. Okay. So good afternoon, everyone. I'm fly all the way here from Melbourne. So right now probably it's midnight over there. So good night from Melbourne. So I just want to quickly do a quick introduction. So some of you may notice that my affiliation is a little bit different from the program booklet. So that's because I recently just moved to the new institution together with my boss. So University of Tasmania, it's my new and current home. So yeah. So today I just would like to share a topic from my PhD, which looks into developing a rapid technique for bacterial detection in foods by using STATIC-HEADSPACE GC by GC. So the story for my work started off with food poisoning, which I'm pretty sure you all are really familiar with what it is. So I'm currently 26, so I ran to the toilet a few times due to diarrhea, so I'm pretty familiar with this. So let me just quickly explain. So basically food poisoning, it's a scenario where you've eaten some foods that's contaminated with foreign materials. So the source can come from many, but the most common one is bacteria. So in the present days, we have lesser incidences of food poisoning, and that is because we nowadays have much more and vigorous legislation around testing. However, the current approach standard that is based on testing is still largely based on conventional culture-based techniques, which is also called the click and wait technique. So because there is high demands in the testing, so this has caused delays, and this has indirectly caused companies to be losing money because products are just sitting in cold storage waiting for the results to come back. So they're just losing valuable product shelf life before they even hit the shelf. So I've talked to some of the food scientists in the labs, and they told me that most of the tested samples that they tested actually came back as negatives. So that is why we need to reconsider another approach, which is much more faster and much more straightforward in order to be able to release some of the burden of the food testing lab, plus to speed up the entire food supply chain. So over here, we're proposing a rapid screening technique. So essentially what we wanted to do over here is to provide something fast and easy that's able to give a much more clear indication of which products are clean and which of products are contaminated. So this brings us to the aim of the study, which is to develop a rapid screening technique for detecting bacterial contamination in foods. So the experiment started off with the model food and bacteria. So we need to start somewhere. So the food and bug that we've chosen is UHT milk and E. coli. So first of all, we started off by optimizing the headspace extraction conditions, and we spiked the samples with a single cell per mil of bacteria, which is really, really low concentration, and compared the profiles between the clean and contaminated samples. So the technique over here is that we chose is headspace GC by GC coupled with FID plus quasi-stop-flow modulation. So besides using the instrument, we've also used the standard plate count, which is a click and wait technique for counting our bacteria, and also conduct some the biomarker discovery by using chemometrics and conduct some data analysis after that. So because the overall aim was to develop a rapid technique, so we are also interested to see what is the minimum incubation time that's required for these bacteria to grow up to a certain level where the system can start to pick up the contamination. So currently the incubation time point that I've chosen sits between 5 to 20 hours. So this is just a snapshot for the conditions of the instrument. So we've used a BP5 analysis as our first column, wax column as our second column, and use a sampling loop. So the carrier gas is hydrogen, GC oven temperature sits around 50 degrees for 8 minutes. So this is just a very snapshot for the condition. For those of you who are interested, I will put in more details at the end of this presentation. So as a result, the overall time to response sits around 20 minutes for the headspace extraction plus 8 minutes of the GC by GC runtime. So we're able to achieve this very short GC time because backflush was engaged. So I'll explain a little bit more in the following slide. So if you just look at the figures at the bottom, it shows you a very clear indication of how the sequence of the injection was being conducted. So you can see from, oh, sorry, let me just put out the laser. So if you see here, for the first sample, it only take the first sample a full 36 minutes. And after that, it basically will just be continuous 8 minutes for the GC injection. So because that, I'm really blessed to have an automated headspace sampler in the lab, because I can just load everything on top of the headspace sampler, program the instrument and walk away. And when I come back, everything will be ready for me for the data analysis. So it's really nice and comfortable for me. So moving on, we have our chromatograms over here. So the left side is our control and the right side is our, left side for you guys, right side for me, for the control and spike samples. So based on this visual observation over here, you can see that, okay, just looking over here, you can see that, oh, these two chromatograms are different. So you can definitely tell that there's something going on. So I'd just like to explain over here why did we just go about and have 5 minutes for GC run. Why not longer? So that is because that we did run a full series of close to 1 hour for the GC. But after that, we discovered that half of these features actually was located in the first 5 minutes of analysis. Because our, once again, overall aim was to develop a rapid technique. So at that time, we're just curious to see whether if we're able to replicate something that is similar if we just look at 5 minutes. So in the end of the day, this is what we got. We just deliberately cut down the GC time to 5 minutes, and this is what we get. So it's quite a huge success for us. So moving on to the incubation time. So for the 5 and 10 hours, we don't really see much difference in terms of the two groups. However, when we move on to the 15 hours, you can start to see the two groups start to separate from each other. And following to the 20 hours, we can see two of the groups. We can see a very clear separation between them. So looking a little bit closer into the compounds, there are 3 compounds in particular. It's the aldehyde, 1-propanol, and ethanol that could be used as an indication for bacterial presence. So if you look at the peak areas over here, it's sort of like self-explanatory. So if you look at the numbers over here, it's actually quite a big gap in terms of the controls and the spike samples. So this could link back to other chromatograms. So why is it so obvious when we look at the E. coli samples that has like a big spot in the chromatograms? So I would just like to quickly summarize the technique that we're using over here. So compared with the conventional culture-based method, with our proposed technique over here, it's able to successfully reduce the overall time to response by a day, so which is a huge cut in terms of time. So this is just a screen capture for the paper that's already published last December. So for those of you who are interested to read a little bit more, here's the QR code for it. So feel free to just scan it and have a read to it. And before I quickly wrap up my presentation, I would just like to give a big shout out to my new supervisor, Dr. Samantha Sawyer from my latest institution, Deakin and the UTAS Scholarship, Dr. Jim and Dr. Rhonda from their helpful guidance for the optimization. Last but not least, Trajan Scientific and Medical for their generous support in the study in order to make this a successful one. And thank you very much.